Yeah. Today, we're cracking open one of the most fundamental processes on the planet, photosynthesis.
Right. It's how life fuels itself.
Think of this as your rapid access guide.
Exactly. And you've given us some fantastic material really spanning the whole range from, the basic,
Textbook stuff.
yeah, right up to the really cutting edge quantum biology. So our mission is to pull out the most crucial and frankly the most fascinating insights for everyone listening.
Okay, so let's start there. At its core, what is photosynthesis? Give us the nutshell version.
Okay. Simplest terms is how plants, algae, and some bacteria take light energy, usually sunlight,
Right.
and convert it into chemical energy. Sugars, basically. It's how they make their own food.
The ultimate solar power system.
Right.
And where does this magic happen?
Mostly inside plant cells in these little structures called chloroplasts.
Chloroplasts. Okay.
And inside those, you've got two main stages happening. First, the light-dependent reactions.
Makes sense. Needs light.
And second, the Calvin cycle.
Okay, let's tackle the light-dependent ones first. You said they need sunlight? Where exactly?
They happen in these internal compartments within the chloroplasts called thylakoid membranes.
Thylakoid membranes, like little sacs inside?
Sort of, yeah. Think of them as the solar panels where that initial energy capture really kicks off.
So what's the play-by-play there? How does it work?
Well, first step is absorbing light energy. That's chlorophyll's job.
The green stuff.
The green stuff, exactly. It grabs that sunlight, and the energy excites electrons inside the chlorophyll, bumping them up to a higher energy level.
So the green we see is actually, it's these tiny energy collectors working away. What happens to those energized electrons?
Next up is water splitting. Photolysis is called.
Photolysis, okay.
That energy from the excited electrons is used to split water molecules, HO.
Right.
And that gives you three things: oxygen, protons, HO, and more electrons, E.
Hang on. The oxygen from splitting water, that's the oxygen we breathe.
That is exactly where the oxygen we breathe comes from, yes. It's a direct product of this step, a crucial link.
Wow. Okay, so we have oxygen, protons, and these electrons that got freed up. Where do they go?
They enter the electron transport chain, the ETC.
Sounds like a tiny production line.
It kind of is. These high-energy electrons move through a series of protein complexes, like passing a baton.
Embedded in that thylakoid membrane.
Right, in the membrane. And as they move, they lose a little energy at each step.
Okay.
And that released energy is used to actively pump protons, those H ions, from the outside fluid, the stroma, into the inside of the thylakoid space, building up a concentration.
So you're creating like a pressure difference, like charging up a tiny battery.
That's a perfect way to think about it. You're building a proton gradient, storing potential energy.
And how does the cell cash in on that energy?
Those protons want to flow back out, down the gradient. They flow back into the stroma through this amazing enzyme called ATP synthase.
Ah, ATP synthase. I've heard of that.
And that flow of protons literally powers it, like water through a turbine, to make ATP adenosine triphosphate from ADP.
ATP, the energy currency for like everything in the cell.
Exactly,
Yeah.
Universal energy packet. And at the same time, those electrons, after going through the ETC and getting another light boost at photosystem I...
And a boost.
Yeah, another hit of light energy, they're used to turn NADP into NADPH.
Okay, so ATP and NADPH, those are the outputs of the light reaction,
Those are your energy carriers, loaded with chemical energy, ready for phase 2.
which is the Calvin cycle, also called the light-independent reactions, but they still need the ATP and NADPH from the light part.
They're absolutely dependent on them. Can't happen without them. The Calvin cycle happens out in the stroma, that fluid-filled space.
Yeah.
And its main job is taking carbon dioxide from the air and using that ATP and NADPH energy to build glucose, sugar.
So grabbing gas from the air and making food, how does that cycle work?
It starts with carbon fixation. There's this really important enzyme, rubisco.
Rubisco. Okay, key player.
Massive player. It grabs a coeuro molecule and attaches it to a 5-carbon molecule called RuBP.
Ribulose bisphosphate.
That's the one. This makes a temporary unstable 6-carbon molecule, very short-lived.
Unstable, like it breaks apart immediately.
Pretty much instantly, yeah. It splits into 2 molecules of a 3-carbon compound, 3-PGA.
3-phosphoglycerate. Okay, so 1 Coro plus 1 RuBP makes 2 3-PGAs. What's next?
Now the reduction phase. This is where the ATP and NADPH jump in.
Time to spend that energy we just made.
Exactly. They provide the energy and the reducing power to convert those 3 PGA molecules into a different 3-carbon molecule, G3P.
G3P, glyceraldehyde 3-phosphate.
You got it. And G3P is a higher energy sugar phosphate. It's the actual building block the plant uses.
Building block for glucose.
For glucose, yeah, and other carbs. Some G3P leaves the cycle to make sugars for the plant's energy or growth.
But not all of it.
No, because you need to keep the cycle going. Most of the G3P stays in the cycle.
Ah, right, to remake the starting molecule.
Exactly. The remaining G3P goes through a complex series of reactions using more ATP.
More energy input.
To regenerate that starting 5-carbon molecule. RBP. So Rubisco has something to grab the next QO with.
So the cycle continues. Okay, let me recap. Light reactions.
Yeah.
Use light, split water, make oxygen, ATP, and NADPH.
Yep.
Calvin cycle. Uses the ATP and NADPH, fixes co-euro, makes G3P, which becomes glucose, and regenerates the starting molecule.
You've nailed it. And the outputs are fundamental. Oxygen for us to breathe, glucose to power the plant, and, well, pretty much the entire food chain.
It really is the foundation. Okay, solid base established. But I know you looked at stuff that goes way beyond this. Quantum mechanics, molecular details, you promised aha moments. Let's dive deeper.
Let's do it. So back to that very first step, light absorption. Remember photosystem 2 and chlorophyll grabbing a photon?
Yeah, exciting the electron.
Well, it's not just a simple energy transfer. There's this quantum effect called quantum coherence.
Quantum coherence. Okay, this sounds advanced.
It is pretty wild. At this tiny scale, electrons act like waves, not just particles.
Wave-particle duality.
Right. So an excited electron's energy can sort of explore multiple paths to the reaction center simultaneously.
Simultaneously, like it checks all the routes at once.
In a quantum sense, yes. It finds the most efficient path almost instantly. It's way faster and more efficient than classical physics would allow. Like having, I don't know, a quantum GPS for energy.
That's incredible. Nature using quantum tricks for efficiency? What's the payoff?
Near 100% efficiency in transferring that initial light energy. It's why researchers are looking at this for, better solar cells.
Makes sense. Okay, so the energy finds its way super efficiently. Then what?
Then the electron gets handed off to a molecule called plastoquinone, or PQ, and that kicks off the electron transport chain.
Got it. And the water-splitting photolysis, you mentioned the oxygen evolving complex, the OEC. Tell me more about that.
Right. Inside photosystem 2, the OEC is the specific site where water gets oxidized, broken down.
To replace the electrons chlorophyll lost.
Exactly. And that's where the protons for the gradient and, crucially, our oxygen come from.
What's actually doing the splitting in there? What's the mechanism?
It's a cluster of manganese and calcium atoms, and neuros usually, held in a very precise protein scaffold.
A specific arrangement of metal atoms.
Yes, and that precise structure is the catalyst. It cycles through different oxidation states to pull electrons off water one by one, incredibly efficiently. It's amazing molecular machinery.
Truly intricate. Okay, then the proton gradient builds up, and ATP synthase makes ATP. You called it a rotary engine.
Yeah, it really is, as protons flow back through ATP synthase driven by that gradient.
Downhill, energetically speaking.
They cause a part of the enzyme, the faux part in the membrane, to physically rotate, spin.
It actually spins.
It's like a tiny turbine, yes. And that rotation is mechanically coupled to the other part, F1, which sticks out into the stroma.
And the spinning makes ATP.
The spinning causes conformational changes in F1 that force ADP and phosphate together to make ATP. It converts the energy stored in the proton gradient into the chemical bond energy of ATP, a molecular motor.
A literal spinning machine making energy money. That's astounding.
Okay.
And after PSII, electrons go to PSI for another boost.
Right. Photosystem I absorbs more light, re-energizes the electrons to an even higher level.
Why the second boost?
They need that extra energy for the next step. Making an ADPH. Those high-energy electrons from PSI are passed to an enzyme.
Ferredoxin NADP plus reductase.
That's the one, FNR. And it uses those electrons to reduce NADP to NADPH.
Which is the other key energy carrier for the Calvin cycle.
Exactly. NADPH carries high-energy electrons. It's reducing power needed to build sugar.
Okay, let's zoom back into the Calvin cycle. You mentioned subphases. Carboxylation first.
Carboxylation, that's step one. Rubisco grabs code and attaches it, carboxylates RuBP.
Making that unstable 6-carbon thing that breaks.
Into 2,3 PGAs, yep. Rubisco is the gatekeeper, bringing atmospheric carbon into the biological world.
And then the reduction phase, using the ATP and NADPH.
Right. ATP provides energy, NADPH provides the electrons to convert 3-PGA into G3P.
The higher energy sugar phosphate.
The actual output product that can be used to make glucose, so energy input here to reduce the carbon compound.
And finally, regeneration, getting back to the start.
Gotta remake that RoBP.
Yeah.
This is where most of the G3P goes. It's a complex shuffle.
Like rearranging building blocks?
Exactly. A series of enzymatic steps using more ATP to turn those 3-carbon G3Ps back into the 5-carbon RoBP. Ready for another round.
It's a true cycle. Okay, but the sources also mentioned issues like photorespiration. What's that?
Yes, Rubisco's flaw. It's not perfectly specific.
Meaning?
Sometimes, especially when co-Euro levels are low and oxygen is high, Rubisco mistakenly grabs an oxygen molecule instead of CoO.
Oh, what happens then?
It kicks off a wasteful process called photorespiration. It actually consumes energy, releases already fixed carbonous co-Euros, and makes toxic byproducts. It basically undoes some of the work of photosynthesis.
So it's inefficient, a problem for plants?
It can be, especially in hot, dry conditions, which is why some plants evolved workarounds.
Like C4 photosynthesis. You mentioned corn, sugarcane.
Exactly. C4 plants have this clever anatomical adaptation. They do the initial co-RO capture in one cell type using a different enzyme, PEP carboxylase.
Which doesn't bind oxygen.
Correct. It only grabs co-RO. It makes a 4-carbon compound, hence C4.
Okay.
That 4-carbon compound is then shuttled to different cells, bundle sheath cells, deep inside the leaf.
Away from the oxygen.
Well, the co-Euros is released there, concentrating it right around Rubisco. So Rubisco mainly sees co-grow, not oxygen. It minimizes photorespiration. It's a spatial separation strategy.
Very smart. And CAM plants, cacti, succulents.
K is crassylation acid metabolism. They use the temporal separation, time-based.
How does that work?
They live in really dry places, right? So opening their leaf pores, their stomata, during the hot day means losing tons of water.
Makes sense.
So they keep stomata closed during the day. They open them only at night.
When it's cooler, less water loss.
Exactly. They take in cocoa at night and fix it into organic acids, storing it.
Storing the CoAs overnight.
Then during the day, with stomata closed tight, they release that stored cocoa internally, right where the Calvin cycle happens with Rubisco, using the ATP and NADPH made from sunlight.
Wow. So they separate co-coker and the Calvin cycle in time, night and day.
Ingenious adaptation for arid environments.
It really shows how evolution tackles these problems.
Yeah.
You also mentioned quantum tunneling, another quantum effect.
Yeah, another one. So electrons moving through the electron transport chain.
Between those protein complexes.
Sometimes the gap between molecules is tiny, but there's still an energy barrier. Classically, the electron needs enough energy to jump over.
Right.
But quantum tunneling says there's a chance, a probability, the electron can just appear on the other side. Pass straight through the barrier.
Even without enough energy to go over.
Exactly. It's like taking a shortcut through a hill instead of climbing over it. It speeds up electron transfer, making the whole chain more efficient.
Mind blown again. Okay, and non-photochemical quenching, NPQ. That sounded like a safety mechanism.
It is. Sometimes, especially in bright light, plants absorb more light energy than they can actually use for photosynthesis.
Too much of a good thing.
Can be. Excess energy can create damaging reactive oxygen species. So NPQ is a way for the plant to safely dissipate that extra energy as heat.
Like a release valve.
Exactly. Protects the photosynthetic machinery from getting fried by too much light.
Okay, this is getting seriously detailed. Fantastic. But you said we could go even deeper. Molecular intricacies, emerging stuff. Let's hit that next level.
Okay, full deep dive. Let's push into the nanoscale structure and function, that quantum coherence in the antenna complex.
Where the light is first captured.
Right. We said the energy explores paths simultaneously. The underlying quantum state is actually a superposition.
Superposition. Like Schrodinger's cat, kind of?
Sort of analogous, yeah. The excitation energy isn't just on one chlorophyll molecule. Its quantum state is spread, probabilistically, across several molecules at once.
A delocalized wave of energy.
In essence, yes. This allows the system to sample the energy landscape incredibly quickly and funnel the energy downhill to the reaction center with almost zero loss. It's exploiting quantum mechanics for ultimate efficiency.
Unbelievable. And the quantum tunneling again for electron transport.
Yes, across those protein complexes. The distances are tiny, nanometers or less. Tunneling allows electrons to hop those gaps much faster than if they had to overcome classical energy barriers. It's crucial for the rapid turnover required.
So quantum effects are not just theoretical footnotes. They're baked into the operational speed and efficiency.
Absolutely fundamental. Now let's look closer at those proteins. Photosystem 2 and its OEC, the water splitter.
The manganese-calcium cluster.
That cluster's precise 3D structure is everything. It cycles through 5 oxidation states, S to S. Think of them as steps.
Steps in pulling electrons from water.
Exactly. Each step involves removing an electron, and often a proton, from bound water molecules. It's the final S to S transition that releases the O Euro molecule.
And the structure holds everything stable during this tricky chemistry.
Precisely. It stabilizes highly reactive intermediates that would otherwise be damaging. It's nature's catalyst perfected over billions of years.
Amazing. And plastiquinone, PQ, the mobile carrier. More than just a shuttle.
Oh, yeah. As PQ picks up electrons from PSII, it also picks up protons from the stroma.
Right.
Then, as it moves across the membrane and delivers electrons to the next complex, cytochrome B6F, it releases those protons into the thylakoid lumen.
So it actively pumps protons too.
Yes, it couples electron transport with proton translocation, a dual role contributing significantly to that proton gradient.
And the cytochrome B6F complex itself.
It's the bridge between PSII and PSI passing electrons along, but it also has its own proton pumping mechanism called the Q cycle. Very efficient. It further boosts that proton gradient,
Pumping even more protons in.
making the gradient steeper, which means more power for ATP synthase. Then plastocinin, a little copper protein, ferries the electrons over to PSI.
For that final energy boost and NADPH production via FNR.
Exactly. The whole chain is a finely tuned sequence of redox reactions and proton movements.
Okay, back to ATP synthase. That spinning motor, more details on the rotation.
Right. The faux part in the membrane has channels. Protons flow through.
Down the gradient.
And the flow exerts torque, causing that central rotor part of faux to spin, like wind turning a windmill.
And that spin is transmitted.
To the F1 part, the catalytic head in the stroma. It has 3 active sites. As the rotor spins, it forces these sites to change shape.
In a cycle.
Yes. One site might bind ADP and phosphate, the next forces them together to make ATP, and the third releases the newly made ATP. Each 120-degree turn produces an ATP molecule.
With incredible efficiency.
Near 100% energy conversion from the proton flow to the chemical bond. And yeah, recent work suggests the rotation isn't always smooth. It can pause or speed up.
Stochastic rotation.
Depending on how much ADP or ATP is around. It self-regulates based on the cell's energy needs. A smart nanomachine.
Just phenomenal. Okay, Talbot cycle again. Rubisco slowness. People are trying to fix it.
Big time. Rubisco is notoriously slow. Maybe only fixing a few co-Euro molecules per second. It's often the bottleneck.
And it grabs oxygen by mistake.
Right. So huge efforts in synthetic biology are trying to engineer faster Rubiscos, or ones that are much better at distinguishing co-Euro from overo, or even design completely new carbon fixation pathways.
Turbocharging photosynthesis.
That's the dream. Imagine crops that are significantly more efficient.
And the regeneration phase. You said it was complex.
Very. It's not just reversing the steps. It involves a whole network of enzymes like transcatallase and aldolase, shuffling carbons around. Think like 3 carbon and 6 carbon sugars being cut and pasted to make 5 carbon sugars.
A molecular jigsaw puzzle.
A highly regulated one. The plant needs to balance the flow through the cycle, what's called metabolic flux.
Adjusting on the fly.
Based on light levels, co-Euro availability, temperature, it has to keep everything in balance to work optimally. Sending just enough G3P off to make sugar, regenerating just enough RuBP.
It's dynamic control. And C4 and CAM, we see them now as evolutionary solutions to these very problems.
Exactly. C4 tackles photorespiration with spatial separation. CAM tackles water loss with temporal separation. They show how the basic C3 pathway can be modified and augmented for specific challenges.
Which inspires bioengineers now.
Yes. Trying to put C4-like mechanisms into C3 crops like rice or wheat is a major goal for improving yield and efficiency, especially in a changing climate.
Incredible journey. From basics to molecular machines. But okay, for the listener who wants the absolute frontier, highest level, let's go there.
Right. The deepest dive. Back to quantum mechanics. That coherence in the antenna complex. It's not just about finding a path. It's about the wave-like nature of the energy, enabling it to effectively sample all paths simultaneously through superposition.
The energy exists as a wave spread across molecules.
Its quantum state does, yes. This allows it to find the global minimum, the most efficient energy transfer route almost instantaneously. It's quantum search in action.
That's just fundamentally different from how we think about things classically. And tunneling.
Quantum tunneling is essential for electron transfer speed. Without it, electrons would be stuck far more often by energy barriers between redox cofactors in the protein complexes. Tunneling provides a quantum leap shortcut.
Making the whole electron transport chain fast enough to keep up with light absorption.
Exactly. Now, the OEC structure, that MUO cluster, its precise geometry isn't just important, it's critical for the water-splitting chemistry.
The S states, S to S here.
Yes. Each transition involves subtle rearrangements of electrons and protons, perfectly orchestrated by the metal cluster and surrounding amino acids. The geometry stabilizes those incredibly reactive intermediate states of partially oxidized water. It's atomic precision.
Preventing chaos at the molecular level. And cytochrome B's proton pumping.
The Q-cycle mechanism is complex but highly efficient. Plastocrenone binds, transfers an electron, releases protons, gets re-reduced, picks up more protons. It doubles the proton pumping for each electron passed compared to simpler carriers, maximizing that gradient.
Every step seems optimized for energy conversion.
Yeah.
And ATP synthase nanomechanics.
The faux rotor, spinning in the membrane, driven by proton flow. The transmission of that rotation via a central stalk to the F1 catalytic head.
Forcing conformational change.
Exactly. Binding change mechanism. The 3 catalytic sites cycle through loose, binding ADP plus pi, tight, catalyzing ATP formation, and open, releasing ATP states, driven purely by that mechanical rotation.
And the stochastic part means it responds instantly.
Responds to the proton gradient strength and the availability of ADP and phosphate, and the back pressure from ATP concentration. It fine-tunes ATP production moment by moment.
Okay. Synthetic and bioengineered photosynthesis. What's the ultimate goal there?
For synthetic carbon fixation, it's about designing pathways, maybe using engineered enzymes like those turbocharged rubiscos that are faster and more efficient than the natural Calvin cycle, potentially bypassing photorespiration entirely.
And artificial photosynthesis.
That aims to mimic the whole process, but maybe more simply, using synthetic catalysts powered by light to split water and reduce CoO directly into fuels like hydrogen or methanol, creating solar fuels.
Using biology's blueprint, but maybe with more robust materials.
Potentially, yes. And for bioengineering, C4 into C3 crops.
Like rice and wheat.
It involves introducing the specialized anatomy, the 2 cell types, and all the necessary enzymes for that CoO concentrating mechanism. It's a huge genetic engineering challenge.
But the payoff could be enormous for food security.
That's the hope. Increase yield, better water, and nitrogen use efficiency.
Wow. We have covered an immense amount of ground from, high school biology basics right up to the absolute cutting edge of quantum biology and synthetic engineering. Photosynthesis is just so much more complex and elegant than I ever realized.
It truly is. It's not just one process. It's this incredibly optimized, multi-layered system operating across scales, from quantum effects to whole leaf anatomy. It uses these amazing molecular machines honed by billions of years.
Running on quantum principles for efficiency.
And it's absolutely fundamental. Without it, life as we know it just wouldn't exist.
So let's leave our listeners with something to chew on. We've seen photosynthesis not just as biology, but almost as nanotechnology, as quantum engineering.
Nature's version, yeah.
So think about this. What new possibilities open up when we stop seeing it just as how plants make food and start seeing it as a blueprint? A blueprint for advanced engineering. Could we harness that quantum coherence for completely new solar tech? Or build catalysts inspired by the OEC to split water with sunlight for clean fuel? What happens when biology becomes the ultimate engineering textbook?
That's a powerful thought. The potential for bio-inspired innovation based on these deep mechanisms is really vast.
Something to ponder. Maybe even explore further in quantum biology or bioengineering research. Thanks for joining us on this deep dive.